type A, B, and O. Keep in mind that in type A, B, and O that we're going to show, it looks like there's a big difference in terms of the physical structure. In reality, there's only one sugar difference. And one sugar can be enough to cause somebody to die from having kind of, somebody introducing blood that is not part, or that the body doesn't recognize. So there's the ABO blood group and the RH. You guys have heard of people being AN positive. The A is part of the ABO blood group, and the positive means that you have what we call a second type of receptor, called an RH receptor. So these blood groups are actually receptors found throughout the red blood cells. So let's take a look at the ABO, all right? Now, before that, understand what is an antigen. An antigen is some kind of protein that is recognized by our body, right? Now, usually the antigen is something larger. Think about when you look at a bacterium. When you look at a bacterium, the whole bacterium is the antigen. Antigens have things sticking out of it, right, that we can attach to. Now, antibodies are what our immune system produces. So then it can recognize the antigen and hopefully destroy it and clear it. So the antigen is a microbe, or if it's a bacterium, will produce antibodies in response to that antigen, the bacterium. The antibodies then attaches to the antigen, and it will then hopefully just, kind of tag it for destruction. So antibodies are something our body produces. Now, antibodies can bind to RBC antigens and result in agglutination or hemolysis. In other words, if we are given incompatible blood, the weird thing about the ABO blood group, let's say I'm type O. If I'm type O, even without a previous transfusion, even if I've never gotten blood from anyone else, if I'm type O, I immediately have antibodies to any type A and type B blood grouping. So it doesn't require a first instance. It's pre-made. After 6 months of life, 6 months is big, right? After 6 months, we start producing antibodies to what we don't have. Now, that's a big thing I want you guys to remember. If you ever get confused, you will produce antibodies to what we don't have. Because if you produce antibodies to what we have, you wouldn't survive, right? Because as soon as you make a red blood cell, your antibody we produced will attack the red blood cell and you would die. So by the fact that we're still alive means that we have antibodies to what we don't have. So if I'm type A right here, that means I have antigen A. Now, antigen A means that on all my red blood cells, I have specific receptors where the combination of sugars allows us to have the A antigen. That sticks out of all my red blood cells. My antibody will be to type B blood then. Makes sense, right? If I have antibodies to A, I would destroy all my red blood cells and I would die. So you make antibodies to what you don't have. Let's say I have type B blood. If I have type B blood, I have a different set of receptors. Now take a look. These receptors look very different from each other, doesn't it? Right? A and B. Now, in reality, I will show you that the only difference is just one sugar. That's it. So in type A antigen, I have this receptor. I have antibodies to what I don't have. I don't have type B, so I have antibodies to type B. How about if I have type B? If I have type B blood, I have the B antigen, which is a little bit different from the A, but it still is different, right? I have the B antigen on all my red blood cells. And then I'm gonna have antibodies to what I don't have. I don't have A, so I'll have antibodies to A. What happens if I'm AB? If I'm type AB, Then I have type A that I have inherited from my mom or dad. And then I have B, I inherited it from the other parents, meaning that I have what's called a codominant trait. A and B are codominant. Keep in mind, when we are conceived, we get 23 chromosomes from each parent. So what happens? Well, I'll have one chromosome from my dad that will code for blood type. And if he has type A blood, he will give me the A type of chromosome. Mom might have type B. Now I inherit one type A from mom, or one type A from dad, and the other type B. Now I have both of these antigens. So both of these are dominant and they're codominant. So I express both of these. Now, these people are usually the lucky ones, right? Why? It's because if I'm type AB, that means all my red blood cells have both type A and type B. Do I make antibodies to them at all? No. The weird thing is, nobody makes antibodies to type O blood. You guys have heard of type O people being the universal donor. There's a reason. And the reason is nobody makes antibodies to type O blood. So for them and their blood is great for the rest of the world, right? The rest of the world can just use their blood type because in their blood, there's no A antigen. There's no B antigen. There's nothing for our antibodies to attach to. So what happens? Let's say I'm type A. I'll have antibodies only to B. If I'm giving somebody that has type O blood, can those B antibodies attach? No, there's nothing attached to. So type O people, their blood is usually the one they're asking for during blood drives, right? Now, specifically type O negative. Type O negative is what we call the universal donor, right? O meaning that they don't have the A antigen or the B antigen. Because they don't have either of them, nobody makes antibodies to them. And nobody makes antibodies to their blood. So if you ever, let's just give you an example. Let's say you're in a car accident. And being part of a really bad car accident, you start to bleed out, right? You have this hemorrhaging occurring. At that point, the paramedics come, right? Well, when you're bleeding out and you're in severe pain and distress, it's a bad time to have to type you and match you, right? There's no time for it. We got to put blood into your body as fast as possible. So what type of blood should I give you then? Type O. Why do I give you type O blood? Because nobody makes antibodies to it. So even if I give you this blood, I don't need to know your blood type at all because I know it should be safe because nobody has antibodies to their blood type. Perfect. Now, what happens when you are type O, right? What happens when you have type O blood? Well, what happens is this. Because you have type O blood, you don't have type A. So you make antibodies to A. Because you have type O, you don't have type B. So you make antibodies to B. So for you that have type O blood, it's the bad part, meaning that you can't receive blood from anybody else. You can only receive blood from fellow type O people, and that's it. So usually what happens is this. You get into that accident. In the field, as you're bleeding, we give you type O blood to make sure you stabilize. As you stabilize, I bring you into the emergency room. And now, because you're stabilized, I can type and match you. In other words, if I'm type A, right here, in the field, no time because I'm bleeding, I'm given type B O blood. But once I get into the emergency room and I've stabilized, then I will switch blood types to one that's specific for me. Instead of giving you type O now in the emergency room, I'll give you type specific blood. Type A blood will be given to me. So then I don't just keep using all the type O. Because if I keep using type O and there's no more type O blood, what happens to the people that get hurt and they have type O? They have no blood to use, right? The only blood that type O people can use is type O blood. If I try to give them A, they have antibodies to A. If I try to give them type B blood, they have antibodies to B. So the only safe blood type I can give to type O people are type O blood. Crazy, right? Now, we call those people universal donors because they can donate to everybody. The bad thing is they can only accept blood from universal donors, people with type O blood. Now, type AB people, they're what we call the universal acceptors. Meaning what? Right? They can accept blood from type O because everybody can. But they can also accept blood from type A. Why? Because they have type A as well in their red blood cells. And because they have type A in their red blood cells, they're not going to have antibodies to A. They can receive blood from type B. Why? Because they have the B antigen as well. Because of the B antigen, they're not going to make antibodies to B. So now they don't make antibodies to A, they don't make antibodies to B, and nobody makes antibodies to O. They can receive anybody's blood. Literally, we can, if you're type AB, right? If you're type AB positive even, you can receive blood from anyone and you should be safe. Assuming that there's no kind of, right, blood-borne diseases, right? But in terms of just ABO and RH blood grouping, you will be safe receiving blood from anybody if you're type AB positive. Now, again, it looks like a big difference, right? Type A, you have the A antigen, you make antibodies to what you don't have. You don't have B, so you make antibodies to B. If you're type B, then you'll have the B antigen. Kind of sticks out, it looks slightly different. Because you have type B, you don't have antibodies to B, or else you never survive. But you do make antibodies immediately after 6 months to the other dominant blood type, A. So you'll have antibodies to A. If you're type AB, you have both receptors, both antigens. Because you have both, you don't have antibodies to either. If you're type O, that means you have neither A or B receptors, A or B antigens. Because you don't have either, you make antibodies to both of the A and B dominant blood types. The good news, if you're type O, you're very wanted by the blood, right? You're very wanted by the blood centers. They will call you almost every day to try to get your blood. The bad news is, if you get into an injured situation where you need blood, the only other person you can receive blood from is type O. The funny thing is this, even though A and B are dominant, O is recessive. That means you gotta inherit the O gene from mom and inherit the O gene from dad. Only when you have both O genes do you have type O. Usually those recessive traits are less common. But what we see is that in our world, actually people that are type O is greater in population, right? It can be between 60 to 70% of people around the world have type O blood, which is good. The rarest blood type is the people with type AB. Now here, they're just kind of showing us what happens in a normal semester, and I think I will see if I can find an actual lab thing that I can introduce to us so that you can see how they're supposed to coagulate, right? Here you have a type A blood donor and antibodies to type B found in the type A blood recipient. So there's a good match, in other words, right? This person that we're giving blood to is type A. Because they're type A, they're gonna have B antibodies. If we correctly read, and now because we correctly read, we're giving them type A blood. Type A blood means it matches. Because there's a match, the antibodies don't attach. Perfect. That's what we should see. Now you would imagine that why would we have a mismatch then if all we have to do is read the history? Well, we do have a mismatch. You guys, when I was starting teaching now it's like 12 years ago, we had this huge issue at one of our hospitals in Milwaukee where somebody was given the wrong blood type, right? The nurse must have been very tired. She gave somebody the wrong blood type just by negligence. And what happens now is that instead of one person asking for your information, they usually have two nurses there, and they always ask for your information and ask if you verify that information. That's what has kind of come about because of that initial mistake. Now here, this is what happens if I have a type A blood donor, and now the person that's receiving it is type B. Obviously, the antibodies for this type B person, well, that antibody is going to be the type A. What happens? The antibody reacts, right, with the red blood cells. As there's a physical match, then the antibodies are going to bind all your red blood cells together and cause what we call agglutination, clotting. As you form that clot forms everywhere where you have blood. So it's going to occur in the brain, causing stroke-like symptoms. It's going to occur in the heart, causing you to have MI-like symptoms. It's going to occur in the lungs, causing almost PE, pulmonary emboli-like symptoms. So this is a whole systemic effect, leading somebody to die an incredibly, incredibly violent, very painful death. All right, now the last thing I'll talk about now is RH. Now again, I wish we'll have more time to discuss this RH and the ABO. Keep in mind with ABNO and with RH, you inherit one of the blood types from each parent. So if I'm saying that, right, if I am type O, and I want you to think of this as you're reading it, if I have type O blood, can I have a parent with type A? Can I have a parent with type B, right? So these are like the Punnett squares, and we'll probably go through that, right? And if we have time after we kind of discuss the RH. So what I want you to think about is, remember, you inherit one of the blood types from one parent. So if one parent has type A, they can give you type A. One blood, and then the other parent gives you type B blood, now you have the AB. What happens if you have one parent give you the type O and the other parent give you type O? You inherited both O traits. You'll have type O blood, right? So in terms of blood type, can you have, let's say you are type O. Can I have a parent that has type A blood? Even better yet, if I'm type O, can I have a parent that has type AB blood, right? And we'll talk about that. Actually, we'll do it right now by going through, and hopefully I can do a sharing screen of the whiteboard. Sorry. For some reason, my whiteboard isn't working, right? Right here. So when we're looking at the whiteboard right here, even though it's not white, and it's not working, where is the whiteboard? All right, well, let me see. Can you guys see the whiteboard at all?
Yes.
Yeah, it's not working for some reason on whiteboard. So I'll just go back to normal screen since it's not working. With the whiteboard, I wanted to show you a Punnett square, but I'll actually have to find a physical whiteboard to show that. And I'll see if I can do that next week. Now, I might have to change next week Tuesday's lecture just because we're having a meeting next week Tuesday as a department at 1 o'clock. So I will probably change it to Wednesday, all right? And I'll send an email out to just kind of remind everybody. Now, with the Punnett squares that I'll show, right? Keep in mind that, and I do want you to play with this, and I want you to think about this. What would happen if I have type O? Is there a chance that one of my parents is type AB? If I'm type AB, is there a chance one of my parents is type O? Right? So this is kind of the way inheritance, and we'll talk about the inheritance hopefully more coming up. Right now, RH. RH is different. RH is a completely different category of receptors. Now, there's like 50 to 60 different type of receptors only in a red blood cell. So we're talking about 50 to 60 different types of, right, inheritance, 50 to 60 type of ability to recognize or incompatibility. So these two, the ABO receptor or the ABO blood group and the RH, they're the ones that are most common. They're the ones that are more likely to cause harm if there's an incompatibility, right? Now, unlike the ABO blood group, there is no dominance. You either have the receptor or you don't. In other words, you're either positive or negative, right? You can't be both. And what we see is this, right? When you're RH positive, say you're A positive, B positive, AB positive, even O positive, right? The positive RH factor is not kind of directed. It can be mutually exclusive, meaning that you can be type O and negative, you can be type O and positive, right? There's no link between those two. So this RH factor is basically an unlinked other receptor found on your red blood cells. Now, ABO, RH are the most important ones because incompatibilities of these can lead to the most kind of danger. Remember, ABO incompatibility occurs without a priming event, right? In other words, if I'm type A, it doesn't require an introduction of type B blood before, right? If somebody gives me the type B blood the first time, I will have antibodies to it already. So that's what we see in ABO blood group. In RH, there is a priming event. It takes a first instance. And when you are RH positive, you don't have to worry, all right? RH positive means that you have the RH antigen, and the antigen is a receptor. So you already have the receptor. So if you already have the receptor, will you make antibodies to it? No, because if you did, those receptors would have been destroyed by our antibodies and you would have died already. So by the presence of you being alive and RH positive, it means that you are not going to have antibodies to these antigens. So being RH positive means you have the antigens, you have the receptors found on the surface of all my red blood cells. Because you have the receptors, you're not going to make antibodies. Perfect. By the way, this is why we call AB positive people the universal acceptors. Because if you have AB positive, you have type A, so I can give you type A blood. You have type B receptors as well, so I can give you type B. And now you have RH as well, so I can give you A positive, B positive, A negative, B negative, O as well. So they can accept anything. The only issue with RH is when you are RH negative. Because you're RH negative, you don't have the antigen. You don't have the receptor. Now, I'm trying to purposely saying antigen receptor right next to each other for a reason, because the antigens are the receptors we're talking about. So you don't have the RH receptor, and because you don't have it, there's a chance of you to form antibodies to it. The biggest difference between RH and ABO is that in ABO, you form antibodies at about 6 months of age, and you continuously form antibodies to what you don't have immediately. Well, RH, when you have an incompatibility, you make antibodies only if you're RH negative and only if you've had a first initial transfusion reaction. In other words, somebody gives you, let's say you're A negative, right? Since I'm A negative and somebody gives me A positive blood, Well, the A's match, so I don't see an immediate reaction. But introduction of that positive blood when I'm negative means that I start forming antibodies, right, to the Rh positive blood. In other words, that Rh negativity, that's the only time when we see an incompatibility, but it requires a first instance. Now, the best example of this is probably the most cruel example there is, and that's a hemolytic disease of the newborn, right? Now, with hemolytic disease in the newborn, what happens is mom is Rh negative, dad is Rh positive. Now, if dad is homozygous for Rh positive, right? In other words, dad has positive Rh from that he inherited from dad, his dad, and then he inherited also the positive blood from his mom. Now he has two Rh positive genes. Well, the only thing he can give to his child is the Rh positive gene because that's the only two he has. So if you have a mother that's Rh negative, that means that she's inherited both traits from her parents. So one parent gives her the Rh negative, the other parent also gives her the Rh negative. Rh negativity is recessive. In other words, you need two of them to actually have that, right? So what we see with the Rh negative mom, her blood lacks this receptor, so there's a chance that we might have an issue, right? Now, if her husband is Rh positive and he's homozygous for it, meaning he has two of the Rh positive genes, because of that, the only thing he can give is a Rh positive gene. All of their kids will then be Rh positive. What happens? Well, with the first fetus inherits the Rh negative gene from mom. From dad, inherits a Rh positive gene. The positive gene is dominant over the negative gene. The baby then will be Rh positive, right? Because it's dominant, all it takes is one of that gene. So dad, homozygous for Rh positive means two Rh positive genes, right? Means that he has nothing to give but the Rh positive blood. Fetus will have Rh positive blood. Mom, Rh negative. What happens? Well, what happens is usually nothing with the first pregnancy, right? The first pregnancy, you guys understand that the only time there's mixing of blood between mom and the fetus, direct mixing, is during vaginal delivery. That's it, right? During normal pregnancy, there's always a little bit of a barrier preventing mom and baby's red blood cells, preventing it from mixing together. But what happens when we give normal vaginal delivery? Well, have you guys ever seen any kind of birthing that's normal vaginal delivery? It's a pretty, I would say, pretty violent process. There's a lot of blood that gets mixed in, right? That's the only time when there's mixing of blood. But let's say Rh negative mom, Rh positive baby, first baby, nothing happens. Because nothing happens, we give normal vaginal delivery, and now there's mixing of blood with that delivery. A little bit of baby's blood is introduced into mom's bloodstream. What happens? At that point, mom starts making antibodies to that Rh positive blood, right? Now, we assume mom and dad stay together, and now they have a second baby. The second baby also then has Rh positive blood. What happens? Well, with the second baby, the mom started making antibodies to that Rh positive blood with birth from the previous baby. Now, the second baby also has Rh positive blood. The antibodies produced by mom from the previous labor and delivery, well, those antibodies start to increase in quantity. The antibody then goes into baby's circulatory system and starts attacking the baby's red blood cells. Those antibodies to Rh attaches to the Rh positive blood in baby, starts forming clots in baby's bloodstream, and eventually causes the baby to die in utero. Before the 1940s, we would see lots of parents, the first baby completely healthy. Every subsequent baby will die in utero, and they would miscarry one after the other every time. And we couldn't figure out why. Because we looked, the ABO blood group matched, right? And why is the baby being attacked then? It was when we found out about this Rh blood grouping that we finally found out that there is an incompatibility issue. Now, can we have this incompatibility issue if mom is Rh positive? No, right? If mom's Rh positive, she has a blood type already. So she's not going to make antibodies. So we're only worried if mom is Rh negative. If you are A negative, B negative, O negative, that's the only time we're worried about an Rh incompatibility. If you're positive and you have ABO, any other blood type, we're not worried about it at all. All right? So how do we prevent subsequent pregnancies from being terminated? So if mom is Rh negative and dad's Rh positive, we know for the fact that, you know, a lot of the kids, a lot of fetuses will be Rh positive. Well, how do we prevent those subsequent pregnancies from dying off? A couple things. First thing is we never allow mom to have normal vaginal delivery. It will be C-section. Not only be C-section, we'll cauterize the blood vessels to prevent introduction of baby's blood into mom's system. That's why we do the C-section. That's number one. Number two, we want to make sure that whatever blood, even that minuscule chance that blood is introduced into mom, well, we want to make it zero. So what we do is give them a shot of something called RhoGAM. RhoGAM contains antibodies against the Rh antigens. Meaning what? Meaning that there might be a couple droplets of baby's blood that's Rh positive. It might get into mom's circulation. By giving them RhoGAM, RhoGAM covers up that antibody, that antigen, covers up the receptor. So then mom's system doesn't recognize it at all. It kind of blinds mom's system, and it tricks mom's immune system into not recognizing that Rh positive blood because there's already something attached to it, right? Now, we do RhoGAM shots with every subsequent pregnancy. So mom has, let's say, 4 kids. First child's okay. We, at birth, for that first child, we're gonna do C-section. Give her a shot of RhoGAM. Second child, do the same thing, right? C-section with RhoGAM. All to make sure that mom's immune system stays naive to it, stays unprimed, so she doesn't form antibodies. Once she starts forming antibodies, let's say you forget to do a RhoGAM shot, right? And a little bit of blood gets in, at that point, it becomes really risky to have that next pregnancy. Kind of crazy, right? Now, in terms of, we're almost done here, ABO and incompatibilities on Rh, the big difference is ABO incompatibility doesn't require that first instance, doesn't require that first child, that first, incompatible blood. You will form antibodies to what you don't have immediately within 6 months of birth. Whereas in Rh, it does require a first instance. Let's say you're type A negative, somebody gives you a type A positive blood. The first time you're introduced with that blood, nothing happens, right? But you start forming antibodies though, after that first instance. So if they try to give you A positive blood again, well, now we have an issue. That A positive blood, the A would match, but the positive trait, right, will cause the antibodies that we had formed to kind of attack the blood right there. Now, let's talk about CBC. Now, on my videos, I have linked It's a little bit better because I'm able to kind of show it physically. And since my whiteboard isn't working here, I can't really do it yet. I'm probably going to purchase another one, a big whiteboard just to show you guys. So in terms of a CBC is a complete blood count, right? The big measurements are for hemoglobin and hematocrit. Hemoglobin measures the amount of hemoglobin, meaning what? Meaning that hemoglobin this is the one I want you to highlight. Right here, underline and highlight it. Hemoglobin measurement is our oxygen carrying capacity. If your hemoglobin levels are low, for males, 14 to 18 is normal. For females, 12 to 16. If they are low, then our oxygen carrying capacity is low. I don't care at that point what my red blood cell count is. Even my hematocrit count doesn't matter. As long as hemoglobin levels are low, you are anemic, right? Anemia can be caused and is going to be measured by your hemoglobin level. Now, usually when your hemoglobin levels are low, your hematocrit levels are also low. What happens? Well, hematocrit is a measurement of the total amount of your blood, whole blood that is made out of cells. And since 99% of your like formed elements, 99% is red blood cells, is a rough indication. The hematocrit count is a rough indication of your red blood cell count, right? In some people, you might have a normal hematocrit count, 44 to 48%, right? You might have a normal hematocrit count. If we look at the hematocrit count, you look like you're normal. You don't have anemia. But in those people, their hemoglobin count is low, right? And when your hemoglobin count is low for any reason at all, we don't even care what your hematocrit and red blood cell count is, right? These can be normal, but as long as your hemoglobin levels are low, you are anemic. Some of you might have heard of pernicious anemia. In pernicious anemia, what we see is very low levels of hemoglobin, but very high levels of hematocrit and red blood cell count. In pernicious anemia, you can make the actual red blood cells, but the red blood cells are mutated and cannot carry, right, and cannot form hemoglobin. Because of that, the red blood cell count, the hematocrit count is normal, but the hemoglobin levels are very low. That's anemia, right? Now, most common kind of anemia is iron deficiency anemia, which most women have. In iron deficient anemia, both your hemoglobin levels are low and your hematocrit levels are low, right? Usually, if you've ever seen anybody that's had to take iron supplements, I remember having to try to do that with my baby daughter, right? Iron supplements suck, right? It's usually this kind of real kind of orangey, dark brown color, like drink, and you're supposed to put it into somebody's mouth and drink it. It tastes awful, right? So I would say you're better off getting it through diet than having to take a supplement. But if you don't eat red meats, then you might have to take the supplement. Now, WBC count, normal, 5 to 10,000. Right? So again, I will link, and I have links to the WBC, right? There's actually a chart I want you guys to know and to draw and to look at. And it's not something I would test the chart, but I would ask you to know it because you will see it when you're in your clinicals, right? Doctors love using this chart. And the chart has hemoglobin and hematocrit on top, and then white blood cell count on the side. Normal white blood cell count, 5 to 10,000. Now, what we normally see when we do a normal white blood cell count is we should have 5 to 10,000. And then if you are getting the sniffles or you're sick, I'll do a CBC with differential, right? These are the normal differential. So if you're completely normal, completely healthy, you're just there for a normal physical exam, right? Yearly physical. I'll do a CBC with differential just for fun, right? And what we'll notice is that your white blood cell count, 5 to 10,000, and your neutrophil count, lymphocyte counts are all normalized like this. What happens if you have an infection? If you have an infection, let's say the infection is caused by a bacterium, right? You have a bacterial infection. You have some kind of, right, pneumonia, bacterial pneumonia. What happens? In your bacterial pneumonia, your total white blood cell count, your total white blood cell count will rise from 5 to 10,000 to 15 to 20,000. That's a normal rise, right? So 15 to 20,000 is kind of really high, but 15 to 19,000 would be something we'd be expected. So now we have an increase in your white blood cell count. With the differential, if it's a bacterial infection, your neutrophil levels will increase from 60 to 70% to 75%, right? It's indicative now that you have more neutrophils. Not only does your white blood cell count elevate, but your neutrophil count increased. And that only happens when you have a bacterial infection. So now I can give you a broad range antibiotic, right? Like erythromycin, like even penicillin, which most people don't even give, right? So augmentin, something like that, something broad range. So I can start treating you immediately for your bacterial infection. Perfect. Now, with coronavirus, what we see is slightly different. In most viruses, we'll see a normal increase in white blood cell count. The normal increase in white blood cell count goes to about 15 to 20,000. When we see that rise, we know you have an infection. When we do a differential with a normal virus, like the cold virus or influenza, what we'll see is a decrease in the neutrophils and an increase in your lymphocyte count, 35%, right? Why do we see an increase in the lymphocytes? We're gonna see in chapter 22, right? We're gonna see that lymphocytes are really good at attacking intracellular type of microorganisms that go into our cells and infects our cells. Well, lymphocytes, when they're attacking a virus, one thing we'll see about a virus is viruses can't replicate by themselves. They're very different than a bacterium. Bacterium can reproduce itself as long as there are nutrients. If there's nutrients, glucose, everything else, they can self-replicate. Viruses can't. In order for a virus to replicate, it has to infect cells. Once it infects the cells, then the virus takes over our DNA machinery and starts reproducing itself. So at that point, we got to attack our infected cells. That's a job of the lymphocytes. The lymphocytes activate because you have a viral infection. Now, instead of 30%, it's at 35%. And now our white blood cell count elevates to 15,000. That means that, right, we have an increased population of lymphocytes. At that point, we know it's a viral infection. In fact, antibiotics will not work. So we will not prescribe antibiotics. Now, if you're sick and you're within 24 hours of showing flu symptoms, right? Within 24 hours, I can give you antivirals, Tamiflu, something like that, right? Indivior, something that allows us to decrease the length of your illness. But they only give those medication within 24 hours, right? We definitely do not give antibiotics if your lymphocyte counts are elevated and your neutrophil counts are low, right? Why? If the lymphocytes are increased and the neutrophils are low, is a sign of some kind of viral or fungal infection as well, right? Antibiotics does no good at that point. So by looking at the differentials, we can have a rough general inclination whether your illness is caused by bacterium or virus. And now we can give you some kind of medications to hopefully, right, decrease the span of your illness. Bacterium, I'll see increased neutrophils, increased total white blood cell count. What happens? Well, we know it's bacterium. I'll give you broad spectrum antibiotic, right? Maybe even a, Keflex or some kind of, right, cephalosporin, right? Now, if the neutrophil levels are low, but my lymphocytes are higher, we know it's probably viral. So if it's within a couple days, I can give you an antivirus, right? Antiviral medication. I definitely don't want to give you an antibacterial, right? Antibiotic. Any questions, guys? Now, keep in mind, right? We will also culture to make sure we can find out which kind of bacterium we are seeing. Once we culture it, now we can get a better indication of the bacterium type and what that bacterium is sensitive to, which antibiotics. So then I can then change antibiotics to something more selective, something that is more likely to kill the specific bacterium found in your body. But usually, right, that takes about 4 to 5 days for the culture. Sometimes if we're lucky, 2 to 3 days, right? Most people don't want to be ill that long, so we give them broad spectrum antibiotics until the culture comes back, the results of the culture. Now, the worst signs that we see is number one, somebody's really ill, somebody has a high fever, and their total white blood cell count doesn't change, right? If you're that ill and your total white blood cell count stays in a normal range or even drops, is a sign that your body cannot mount an immune response. It cannot fight that infection. That's a severe bad sign, right? That's what we see with people that are elderly that are getting coronavirus, is that we see their white blood cell count basically in a normal range and no increases in the lymphocytes, right? And in some of them, there's actually a decrease in the lymphocyte count. That's a sign that this is a bad indication that person might not survive because they can't mount a sufficient enough immune response. They can't activate the white blood cells, those lymphocytes that are meant to fight off that viral infection. Right. Now, we will talk about blood chemistry. Some of you might watch like Grey's Anatomy, right? Or, any of the medical shows, and they talk about a CBC with diff. All right, and then right after it's hit, they want to do a BMP or Chem 20. BMP Chem 20 is a blood chemistry, right? BMP is basal metabolic panel, and it measures your sodium, potassium, your bicarbonate. It measures all those ions, right, that are found in your plasma. So then we'll see, well, does this person have an imbalance in potassium, calcium, that's causing some of the symptoms, right? That's what the BMP or Chem 20. Chem 20 is a little bit, less kind of things we're testing for. It's the 20 highest kind of solutes, whereas the BMP tests everything. Right. Any questions? I don't think we are going to get to chapter 20. I will instead start talking about chapter 20 next week. Now, I do have that meeting next week, and I want to ask